Good evening to everyone. Today we'll learn something about the structure and functions of the muscle tissue is concerned. And everyone now that muscle tissue is also one of the important connective tissue of the body. And we need to learn this as in the class 7 of veterinary anatomy, physiology and biochemistry. So the muscle is derived from the Latin word musculus means the little mouse. So it is named because some muscles like biceps pop up as the mouse is dashing under the skin. And there are three distinct type of the muscle. Number one, skeletal muscle, cardiac muscle, and the smooth muscles. Whereas the skeletal and cardiac muscles uh, are referred as the striated muscles, whereas the other muscles are called as the transverse binding pattern. And the skeletal muscle is also referred as voluntary muscle, while smooth muscles, most of the smooth muscles and cardiac muscles are called as involuntary muscles because these are not under the control of anyone, whereas the skeletal muscles, except the diaphragm, etc., they are under totally the control of a person. And all activities that involve movement depend upon the muscle. In case of a human body, there are 600 muscles, and various purpose of the muscles are number one, the locomotion, upright posture, balancing on two legs, because human is a person who walks on two legs, and animals walk on four legs, and some animals do also walk on two legs like the poultry. So the support of internal organs is also another responsibility of the skeletal muscles. Then controlling valves and body openings and production of the heat and movement of the materials along with the intestinal tubes. So history about the muscles, how they are developed, etc. Then this Leeuwenhoek, Anthony von Leeuwenhoek in 1680 first observed muscle striation, means he noted that they have got the different type of the bands and fibers. And Wilhelm Pune in 1864 purified myosin from the muscle, and Leon Frederick in 1864 a bands and eye bands based on the blueprint, and said a band does not change during the contraction, but eye band becomes smaller. Before 1954, muscle contraction was believed to be by folding or coiling up of the myosin by soluble ATPase enzyme. Means very recently, about 50 years back, that's all. So the Vladimir and the Militsa in 1939, they said myosin is the ATPase, whereas Albert Sen in 1942 discovered actin and activator of the myosin filament. Andrew Flukeley in 1960 explained about sliding filament history of the skeletal muscles. Like this, they explain the skeletal muscle development. Initially, the muscular system is also known as the mesoderm, and the example of this one are the iris muscle, then neuroderm, especially the muscles of smooth muscles, especially. Then paraxial mesoderm is also known as leading to the somites, then the ventromedial or scleroderm, and it's adhering to the vertebrae plus ribs, and dorsolateral, that's also known as dermomyotome, etc. So the somatic or the skeletal muscles do contain different structures as depicted in the photo here, like the vertebral column contains the this neural tube, and the other things like the umbilical vesicles are there, or yolk sac in case of a fetus, etc. So this is the one how the skeletal muscles do develop from the childhood. Then apart from this one, there are the terms called as the myotomes. And once again, different type of the structures are there in different bodies, like short muscles, long muscles, then the psoas muscles, stomach muscles with smooth muscles, then rectus abdominis muscles. Different type of the muscles are there, like the muscles of the pharynx, then myoblast, occipital, myotome, and the muscles of the tongues. All these are called as the smooth muscles. So most of them are involuntary in nature. So three main muscle types as depicted earlier, smooth muscles, or they are all called as the involuntary muscle system. Then cardiac muscles also involuntary, whereas skeletal muscles do have the striated cylindrical, then multifunctional fibers. They are parallel to each other. So these are voluntary in nature. Both the cardiac and smooth muscles are having the involuntary in nature. Then skeletal muscles. So this is a cross section of a skeletal muscle. Then we can find the anatomy like this. The muscle connective tissue. Number one, the tendon is being connected with a bone by the connective tissue tendon, and the muscle bundles are there. Then the capillaries, which are supplying the blood to the skeletal muscles, are always required. Then myofiber or muscle fibers are very much required, and of course, both nerve supply, blood supply is also there. And in this outer coat is called as the epimysium. Then inside that thing, both these muscle layers are there, or the endomysium is in between the fibers. So these are all called as the individual muscle fibers. Then in between these, the endomysium. Endomysium is present just like the sandwiched portion. Then containing many type of these muscle fibers, the perimysium is the one which is having the especially the tissues which are going to covered by one of the sheets. Then the sarcomere, then myofilaments, then sarcoplasmic reticulum instead of endoplasmic reticulum in case of a normal cell. These are called as the sarcoplasmic reticulum. And the fascicle is wrapped by the perimysium. So peri means surrounding. So this is the cross structure. Please remember this cross structure of a skeletal muscle which is adhered to the bone by the tendon, then epimysium, endomysium, muscle filament, blood vessels in between, then fascicle and perimysium. These are all the structures, and one has to remember. So this is a photo of the skeletal muscle fiber. So the bands here, very pink type of the bands, and everywhere the nuclei are located far away from each other. And here you can see the striations in between the skeletal muscle fiber, and there is a connective tissue in between this thing, and these are all the straight muscle fibers, whereas the muscle connective tissue is also called as the epimysium. So this is the connective tissue sheath covering the entire muscle. Epimysium means outer coat, and perimysium, it's a connective tissue sheath surrounding the bundles of muscle fiber. So this is a muscle fiber. The coat of this is called as the perimysium. The endomysium surrounds the individual muscle fiber here. You can see that perimysium, outer coat, the endomysium, a bundle of the muscle fibers are covered by this thing, whereas the endomysium is covering each muscle fiber. So then it's a bundle of entire uh, muscle fibers. Muscle fiber is consisting of the muscle fascicles. Now, the muscle bundle contains 10 to 20 myofibrils, myofibrils, like just the small sizes structures and encased by the perimysium. So this is the cross section of a muscle, and we can see the cross section, especially just like a salad you can see. So the capillaries and capillary endothelial cells are also there in between the individual myofibrils and separated from the myofibril by interstitial space. Once again, so here you can see that the blood vessels are supplied to every muscle bundle. Then in between these nucleus are there, myofibrils are present, then muscle fibers are there, and also the nerve supply is there to every muscle fiber. Then myofiber is also the muscle fiber, which is highly specialized cell and is the structural unit of the skeletal muscle tissue. For example, it constitutes about 75 to 92% of the total muscle bundle. And here you can once again you can see that tapered end of the muscle fiber, then the sarcolemma, the outer coat, and myofibrils are inside. And in between two muscle fibers, there will be a connective tissue which binds them, and the nucleus of every cell is being visible. So, the skeletal muscles are composed of large number of long cylindrical thread-like structures, unbranched multinucleotide cells called as the muscle fiber. So these are the muscle fibers which taper slightly at both ends and are arranged parallel to each other. The fiber measures between 0.01 to zero point 0.1 millimeter, 10 to 100 nanometer in diameter, and are several centimeters long. Several centimeters long. Then myofibril are long, thin cylindrical rods, usually 1 to 2 micrometer in diameter, and they constitute 75 to 85% of the yes muscle fiber volume, and they lie in parallel to form cell, and their long axis parallel to long axis of the fiber. The myofibrils are bathed by sarcoplasm and extend to the entire length of the muscle fiber. And there are two types of the filament within the myofibrils, thick and thin filaments. And these thick and thin filaments are aligned parallel to each other, and these arrangements give a striated appearance. Here you can see that sarcolemma, myofibers, and the connective tissue. Whereas the sarcomere is a band consistent wide like this thing, and one band variable with depend on state of the contraction. Z disk boundary of the sarcomeres, H zone no overlapping thick and thin filaments variable in width. Whereas the fifth is pseudo S zone area of no myosin heads. So here you can see that sarcomere is entire thing is called as the sarcomere, which contains the thin filament and M line and H zone here, and the Z disc which connects each other, and the band I, A band and I band. So these are subsequently they are attached to each other and these are responsible for contraction and relaxation of the muscle fiber. So myofilaments are the basic structure constitute A band, especially myofilaments. So the thin filaments is called they are called as the myofilaments. Then 14 to 16 nanometer in diameter, very small structure, 1.5 micrometer in length, and prominent protein is myosin. And thin filaments 6 to 8 nanometer in diameter, 1 micrometer on either side of the Z disc, and constitute I band and extend beyond the I band into A band and predominant protein actin filament. Then Z duct ultrastructure is made up of Z filaments connected with the filaments on either side of the Z disc, and the one actin filament connects to four uh, Z filaments that pass through Z disc and then connect with the actin filament in adjacent sarcomere. So what are all the proteins of the myofibrin? There are about 20 different type of the proteins where 6 constitute about 90% of the total myofibrin proteins. And myosin, actin, titin, tropomyosin, troponin and nebulin are the 6 constituent of proteins. So on the basis of the function, the actin and myosin are contractile in nature because they expand and reduce in structure, whereas regulatory tropomyosin and troponin are the regulatory one, whereas the cytoskeletal titin, nebulin are integral to the structure of Z disc, and the here you can see the structure of the troponin and tropomyosin and F actin, which is interlocked with each other. So the actin filament is the 20% of the muscle fiber, muscle protein, and globular shaped structure about the 5.5 meet this millimeter, nanometer diameter. These are all the things. And bean type of the shape is there, G-shaped actin, monometric form. And G actin monomers polymerize to form F actin. And two strands of F actin are separately called around one another to form super helix. So there is coiling and the further coiling, and this is called as the super helix structure. And the myosin, this fiber protein, 45% of the muscle protein. So this is called as the meiosis nucleolus, and this is a continuous elongated rod shape with a thickened portion at the one end. This is the one. They are called as the heads, myosin heads. And head region is double headed and projects laterally from long axis of the filaments. So sometimes it is on both sides. Whereas the portion between head and tail is known as the neck. So these are all the tiny structures in between the muzzle, Z disc is there, thick filament, and once again I band, A band, once again band I. So the structure are very much required for the contractile strength of the skeletal muscle fiber, whereas the thick filament is called as the myosin, which contains the tail and also the heads. Here you can see that there are different heads and they together are called as the thick filament myosin, whereas the thin filament is also called as the actin. And this is a portion of the thin filament which contains the troponin complex or tropomyosin, tropomyosin, and G actins, just like the blue bead structures. Then there are certain regulatory proteins like tropomyosin, then troponin, whereas these are also present as one of the constituent components of every muscle fiber. Then cytoskeletal proteins are also called as titin, then the largest polypeptide known, especially the cytoskeletal titins. These are all the titins in between this thing. So, actin thin filament extend longitudinally in each half sarcomere from M line to Z disc, and portion of titin in A band is inelastic and that I band is an elastic. So binds to the outside shaft of thick filament and C protein that encircles and stabilizes the thick filament. And nebulin is containing about 4% of the muscle protein, and it's also one of the important structure. And apart from this one, C proteins are there, H protein, myosin, and many more, alpha, actin, etc. Then here you can see the arrangements of myofilaments in a sarcomere, which contains the thin filament, thick filament, and a combination of thin and thick filaments in a sarcomere. Then the percentage-wise contribution of different myofibril proteins, the myosin is highest, actin second highest, tropomyosin and tropionin are equally contributing, whereas the M protein and C protein are somehow half to each other. Sarcoplasmic reticulum is just like endoplasmic reticulum of a cell which contains a cisternae that form closely meshed network around each myofibrils and T tubules associated with sarcolemma and elements of the sarcoplasmic reticulum. They have the longitudinal tubules, fenestrated collar, then terminated cistern A, and they also contains different other components. Just you can here you can see the fibrils, sarcolemma, then mitochondria are also present, then the nucleus is present. So going more detailed cross section. So this entire outer coat is called as the sarcolemma. Then inside sarcoplasmic reticulum instead of a cell endoplasmic reticulum. This is sarcoplasmic reticulum, and the terminal cisternae are present, followed by T tubule and triad. And here you can see that sarcoplasmic reticulum are surrounding the muscle fibers. Then mitochondria located on sarcoplasm, which are the powerhouse of every cell, including the muscle cells. But very similar to that of many other cells of the body, lysosomes are also small vesicles located in sarcoplasm do contain enzymes capable of digesting a particular cell. Cathepsins are the proteolytic enzyme is a major importance, then followed by Golgi complex and secretory cells. So connective tissue is surrounding muscle bundles and muscle fiber is fibrous. For example, the connective tissue proper, bone cartilage, supportive connective tissue, ground substances do contain the substrates and end products of CT metabolism such as tropocollagen and many more structures. So these are all the extracellular fibers. Then there is the elastin is there. Apart from this one, there are the connective tissue cells are there like mesenchymal cells, adipose cells, fibroblast cells. And the neuromuscular junction is a special structure in between the nerve and muscle fiber in case of the skeletal muscles and do contain the motor end plate. Then pocket formed around motor neuron by sarcolemma. As usual, everyone know that acetylcholine is released from the motor neuron and causes end plate potential or miniature end plate potential. Then depolarization comes and muscle contract. So this is the neuromuscular junction wherein which a motor neuron fiber is ending with that of a muscle and nerve fiber branches here, then these red structures are called as the muscle fiber nucleus. And apart from this one, there are a lot of vesicles which do contain the acetylcholine, and this is called as the motor end plate and folded sarcolemma. And apart from this one, this is responsible by secretion of acetylcholine and acting on the N type of the muscular type of the receptors, which are very specific to the skeletal muscle and the skeletal muscle contract. Motor unit is the single motor neuron and muscle fibers. It innervates eye muscles 1 is to 1, hamstrings 3 is to 1, and many more. So muscle contraction, actin and myofilaments are moving together, and sliding filament theory step one. There are sliding filaments are there because of the entry of calcium, then calmodulin, and acetylcholine is a neurotransmitter in almost all the skeletal muscle fibers, whereas the smooth muscle fibers do contain both adrenaline and also the acetylcholine or norepinephrine. So there are a lot of theories, sliding filament theory 1 and 2, which the calcium bonds to troponin and myosin binding site is being exposed, and hence the contraction of the muscle fiber take place. And sliding filament theory is also known as the step 3 theory, which is uh, also functional in many type of the skeletal muscle fibers. So the filamental theory continues like step 4, and it's also called as the power stroke, which is caused by cross bridge if it is broken. Then there is a one always energy is required in the form of the ATP, which is later converted to the. This is a cascade of events. Whenever the muscle action potential arises from the nerve, then the secretion of acetylcholine happens. This includes the cascade of events. Then there will be increased intracellular calcium, then calcium calmodulin complex will be produced. Then the muscle contracts. Later, it's going to relax. So this in the rest phase, this is the structure. Especially in whenever it contraction, this is the structure in which the contraction stage, the Z disc is contracted much more. Then sliding filament theory or relaxation always happens once the neurotransmitter activity is either reduced or the acetylcholine is broken into acetic acid and choline and diffuses out of the neuromuscular junction. Then, the what is the source of the energy for muscle contraction? As usual, it is adenosine triphosphate. So, what is the sources of the adenosine triphosphate? So, it is a phosphocreatinine, creatine, then glycolysis, then oxidative phosphorylation. They are the important source of adenosine triphosphate. So this is the step how a muscle fiber is being supplied with energy by binding with that of the muscle fiber and initiating a cascade of events. So a glucose comes inside and glycolysis happen. Then glycolysis produce the lactic acid. So the ADP is converted to adenosine triphosphate and the oxygen enters the muscle fiber, then a reaction called as the oxidative phosphorylation takes place. Then fatty acids and amino acids do always required for the muscle to contract. So, the classification of muscle fiber is once again they are subclassified into three different methods: histochemical staining of the myosin (ATP), myosin heavy chain isoform identification, and biochemical identification of the metabolic enzymes. These are all the fiber type of the classification. So the individual skeletal muscle fibers are classified into slow fibers, fast fibers, and another type 2 fibers. For example, these are the slow fibers or slow twitch fibers and slow oxidative fibers. So these are useful for the running, etc. And the fast twitch fibers, their role is very important. And apart from this one, the intermediate fibers and fast oxidative glycolytic fibers are involved. Then fast twitch response of the muscle takes place by several cascades of the events and the opposite to this thing, slow twitch muscles. So slow twitch type 1 endurance and are smaller than fast twitch muscles. So the intermediate twitch muscles are above slow and fast. Then what are all the receptors in case of a muscle spindle? So they have got the dynamic type of the especially so a lot of receptors are going to control the secretion and action of the commonly secreted neuro, especially nerves, especially nerve agents or okay, neurotransmitters. So sensory nerve fiber is going to bind with that of the nerve endings and releases acetylcholine. Then the muscle fiber is having connection with the sensory or afferent means which carries the pain or any other threshold from the particular site of action to the brain. And then later the thing is the Golgi tendon, then alpha motor neuron inhibition, then sensory neurons. They're all the different parts of the structures of a muscle fiber. Then lot of muscle dystrophies are present, and the calf muscle dystrophy, limb girdle, and many more are there. And you can just refer and gain some knowledge of the particular thing is there, and lot of disorders are also present, and this much is enough for the exam is concerned. And if anybody want to learn much more, you can refer the different textbooks which are already mentioned for the particular course is concerned. And of course, the annexures, figures, everything you can study it with the references like the Pasternak and human skeletal fiber type classification, work, and many more type of the Harrison's principles of internal medicine, and many more things are books are read. And for veterinary anatomy books, different type of the anatomy books are available, or veterinary physiology or biochemistry. These are all supporting things from the original author. Thank you. Have a great